All right, guys. So we're gonna give this a shot. This will be our first run. What we're gonna do today is, or in this video, I guess, we'll be looking at the nervous tissue lecture, which is first of our, well, it takes 4 lectures to get through the entire nervous system. 3 of them will be in this unit, unit number 4, and then the 5th unit will have one of the final or the final lecture on the nervous system. So what you guys can see now is hopefully exactly what you guys have there in front of you, either they're printed off or a digital version. What I'm going to do is just go through these just like I have been doing in class, so you can follow along and I'll read through and kind of point stuff out in pictures, and we'll treat this just like we did, when we were still meeting together. So first things first, just kind of introducing the stuff that make up the nervous system. Even though our nervous system is relatively small, I really shouldn't even say relatively, it's rather small, only represents about 3% of our total body weight. It's really complex. Physiologically, it's probably the most complex of the 11 body systems. Anatomically, there are some definite complexities to it, but there's just not as many structures. So those structures that do make up the nervous system include the brain, which is going to contain about 10 billion functional cells called neurons. Attached to the base of the brain, emerging from it are 12 pairs of cranial nerves that, obviously 12 pairs makes 24. The spinal cord is basically an extension of our brain that runs down through our vertebral canal and connects the brain to what we call our spinal nerves. Spinal nerves, there's 31 pairs of those, so 62 total, that serve the left and right sides of our body. And then in close contact with our spinal nerves and our cranial nerves are collections of nervous tissue we call ganglia. The ganglia, typically what you'll see is not necessarily just like a structure that you could point to. There are a couple of instances where that's the case, but typically it's just kind of a cluster of what we call cell bodies of neurons. And as we'll get to this lecture, we'll learn what cell bodies are. The key feature of ganglia is that they're located outside the brain and spinal cord, and as I said, closely associated with cranial and spinal nerves. Other things include enteric plexuses. These are extensive networks of neurons located in the walls of GI tract organs, helping regulate the digestive system. They turn on when the muscles in the stomach wall contract, when to turn your food. We also have, obviously various digestive secreting glands in places like our stomach and intestines. The enteric plexuses turn those on and off to help us digest food. We'll talk more about those really kind of later on this semester when we get to the digestive system. So for now, we'll kind of for the most part, leave it there. And then sensor receptors are the final component, either parts of neurons or specialized cells that monitor changes in the internal external environment. We have lots of them in our specialty sensory organs like our eyes and our ears and our tongue, monitoring those special senses. We've got a lot of them in our skin, detecting changes in temperature and when we're itchy and when we're cold and when we're hot, stuff like that. So lots of sensory receptors located, kind of all sorts of different places in our body. But collectively, those different structures allow us to kind of perform 3 major or just kind of basic functions, and that's what's listed off here at the bottom, what we call the sensory, integrative, and motor functions. First function relates to those sensory receptors, and that's the sensory function, which let's do reading off here. Sensory receptors detect internal stimuli like an increase in blood acidity, external stimuli like raindrop landing on your arm. The type of neurons that serve this function are called sensory neurons, which is handy. Sensory neurons perform the sensory function. You can also call them afferent neurons, and we'll talk about that term here more on the next page. But the function of those neurons is to carry the information into our brain and spinal cord by way of those great cranial and spinal nerves that I was just talking about. The integrated function is probably the most, well, it's not probably, it is the most complex of the 3 functions. This is where we process that sensory information. So, talking about like the raindrop hitting your arm, that signals to the brain, and our brain processes that. We are aware of that. If we're cold, we think of like, shivering as a response. The integrated function is the deciding response, what to do with that sensory information. And in the case of shivering, our brain, where we perform a lot of our integrated functions, is responsible for deciding that we need to shiver so that we heat up our body. So integration refers to processing that information, analyzing, storing some of it, and making decisions for appropriate responses. Now, one important thing about the integrated function is a concept called perception. Perception is described as the conscious awareness of sensory stimuli. Takes place in a region of our brain called the cerebral cortex. We have perception over things like, a lot of which are like external to us, like when someone touches us on the arm, or when we're itchy, or when we're hot, when we're cold, when we feel pain. We are well aware when those things are taking place. We don't have conscious awareness of every sensation. We can't feel when our pH, our blood acidity is changed. We don't know when our PCO2 is, out of whack. So not every sensation is consciously interpreted by us, but our brain typically, which performs the integrated function, kind of takes care of it on its own. Type of neurons that serve this function are called association neurons. Typically, I use the term interneurons. I like interneurons, and you'll see why here later in the lecture. I'll talk about those a little bit later. Motor function is the final functions, part of the nervous system that elicits a response. In that, I mean, like in the case of going back to sweating or shivering, when we're hot, when we're cold, the signal leaves the brain, leaves the spinal cord, it travels out to what we call effectors. Effectors being our muscles and glands, typically. Now remember, there's 3 types of muscles. Skeletal muscle would be the ones that would be working in the case of like shivering. But if we had like a change in blood pressure or heart rate, it could include smooth muscle or cardiac muscle, which are the other 2 types of muscle. And then in the case of like sweating is a result of sweat glands. So those would be the effectors in that situation. Signals carried out by way of those cranial nerves attached to the brain, or the spinal nerves attached to the spinal cord, once that sensory information is integrated. Type of neurons that perform this are called motor, which is nice, motor, or efferent neurons, which cause muscles to contract and glands to secrete. And really, I could say muscles to not contract. So sometimes we want a response to stop muscles from contracting and that kind of stuff, which is pretty far from what we talk about in this class. As far as this picture at the bottom, it's not something I would ever ask you to know. It does, show you those different structures that I kind of was talking about. And it breaks them down into 2 boxes here. On the left, what we call the CNS, and over here on the right is the PNS, which I'll explain what those abbreviations mean right here on this page. Speaking of this page, let's go ahead and dive in on it. The human body has 2 organ systems dedicated to maintaining internal coordination. First of which is something that we're going to talk about in the next unit, the endocrine system, which communicates by sending and releasing chemical messengers known as hormones that are released into the bloodstream, and then elicit a response. What we'll be talking about for this lecture, the next lecture, and the next 2 after that, so 4 in a row, will be the nervous system, which employs electrical and chemical means. Electrical means being action potentials and nerve impulses. Chemical means being what we call neurotransmitters, which send messages very quickly from cell to cell. The cells in the nervous system that use these means are known as neurons. And we've already talked quite a bit about those, the different types, but we'll talk more about those here on the next couple slides. Those neurons that make up our nervous system, billions of them, and then the supporting cells, there's even more of those, what we call neuroglia, are organized into 2 main subdivisions. The central nervous system, which is what the CNS is short for, and the peripheral nervous system, or the PNS, which is what that is short for. Central nervous system, described as the structural and functional center of the entire nervous system, consists of our brain and spinal cord. That's kind of the key thing to know. Aside from what it does, these are the 2 things. It acts to integrate, so once again, to process that sensory information and decide what to do with it. So therefore, it's the command center of our nervous system. Really kind of mainly in charge, brain, and second in command, we could describe the spinal cord. Everything else that makes up the nervous system would be thrown into the peripheral nervous system. So that includes those cranial nerves, those spinal nerves, the ganglia that I mentioned, the plexuses, the sensory receptors, all those different things I'm not going to read through that we were just talking about on the previous page. And that's a lot of stuff. So we actually take the peripheral nervous system and we kind of break it down into some smaller divisions based on who's being regulated. And by who, I mean the effectors. And, there's three major divisions: the somatic, the autonomic, and then the enteric. The somatic is associated with, and a key word here in this sentence is voluntary control, and it is associated with carrying information to and from. Another key set of words here would be our skeletal muscle, and that's what allows you to, wave your hands, write with your pencils as you're taking notes, with a purpose. And we do this on purpose, and that, I mean, I am controlling it. I know that I'm doing it. It's not just being done, outside of my, awareness. Now, as far as that goes, we're talking about signals leaving. So we would call those neurons, motor neurons that conduct the impulses from the central nervous system to the skeletal muscles that I was just mentioning. There's also the sensory neurons, which are going to carry the signals the opposite way from the somatic receptors on our head, body wall, and limbs, and from receptors for our special senses to the central nervous system. We'll talk more about sensory motor here at the bottom of the slide. Now, the next division of the peripheral is the autonomic nervous system. And I always tell students, when you hear the word, and you see the word autonomic, think automatic. And that's because this division of the nervous system is associated with involuntary control. Involuntary means that we really have no conscious awareness. Excuse me, it's done for us. And a lot of times without even our, a complete awareness. The effectors in this are called autonomic effectors. And typically we talk about cardiac muscle in our heart, smooth muscle in places like our blood vessels, our stomach and intestines, the uterus, the bladder, so on and so forth. And then, the glands, which include sweat glands and oil glands, but it could also include glands, inside our body, much more complex than, sweat, oil glands. We could also throw in as autonomic effectors adipose tissue, which we know is fat, as well as other things. But these first three, cardiac and smooth muscle and glands, are typically the three ones that are known the most. And we can subdivide the autonomic nervous system into what we call a sympathetic and parasympathetic division. The sympathetic division is responsible for producing what we call a fight or flight response, enabling our body to either fight or flight. Basically, what we're talking about is just ability to perform physical activity. Now, it can be a life and death situation like running from a bear. We call these like an adrenaline rush. It could just be simple as, running around the building, doing some jumping jacks, whatever. Things that occur when we activate the sympathetic nervous system is increases in heart rate, releasing sugar from our liver into the blood. We'll breathe more rapidly, our blood pressure will go up, we'll increase the amount of blood that flows to our skeletal muscle, which is going to enable it to fight or flight. Because to do either of those things, we have to contract muscle. What's more active probably in you right now, as you're sitting here watching this video, is what we call the parasympathetic division, which produces what we call a rest and digest response. Examples of that would be the secretion of saliva, digestive enzymes in the stomach, maintained heart rate somewhere, 60, 70 beats per minute, breathing rates hopefully not too rapid, not too deep, so on and so forth. The great thing about our body is we can, switch between these two divisions kind of on a flip of a dime, where, you might be sitting around just casually doing nothing. And then, you get up and you go for a run. Pretty much immediately, your body responds by activating that sympathetic division, activating that fight or flight response, which is cool. All right, the third division of our peripheral nervous system, shown over here on the right, what we call the enteric nervous system, associated with the same thing here, involuntary control. And specifically, we're talking about the organs of the GI tract. So we see GI tract, gastrointestinal tract, we're talking mainly about stomach, small and large intestine, but it also, includes a couple other organs as well. Many of the neurons of the enteric nervous system function independently of the autonomic and central nervous systems to some extent, although they also communicate by way of those sympathetic parasympathetic neurons that I was just talking about. In fact, if you kind of think with common sense from what we've just learned, imagine it's not hard to imagine that the parasympathetic is going to help amplify and accelerate activity of the enteric, whereas sympathetic is going to inhibit the activity of the enteric nervous system. Neurons, sensory neurons of the enteric nervous system respond to changes within the GI tract. So like as our stomach fills, it stretches, pH inside changes. So that activates the enteric nervous system. And then the motor neurons are going to be activated to control digestive function by, triggering the release of digestive enzymes and causing muscles to contract and that kind of stuff. So down here at the bottom, the figure that I've already kind of alluded to a couple times, it's kind of a schematic to show the divisions of the nervous system at the top, the central nervous system, and in this big purple box, the peripheral nervous system. What we see in the three sub-boxes of the peripheral are these somatic, autonomic, and enteric that we just talked about. We also see arrows in different colored boxes, either pointing to or away from the central nervous system. You think about these different divisions of the peripheral nervous system, somatic, autonomic, enteric, think of them each like a two-lane highway, where there's signals going in one direction and signals going in the opposite direction. We always describe where things are going in relationship to the central nervous system. So in relation to the brain and spinal cord, if the signals are going towards it, we call this the afferent division. Also, we can use the term sensory if we want to. And if you go back to that previous page, we said sensory afferent neurons provide us with the sensory function. So those terms are both synonymous with the incoming information. Whereas the efferent division or motor pathway, which is shown here in red, involves the signals leaving the brain and spinal cord, going away from. And as I just showed you on the previous page a couple minutes ago, the motor response or the motor function of the nervous system was performed by either motor or, as we can call them, efferent neurons. So now you see why those terms are what they are. Figures, just something to kind of help explain and imagine what's going on with the nervous system, but it's not something that you would want to study or focus on. Okay, now time to talk about neurons, which we've been talking about since that very first slide. Other term for neurons are nerve cells, which we can describe as excitable. And excitable is a term that means the ability to respond to a stimulus and convert that signal into an action potential. So they're excitable cells that conduct the impulses that make possible all those nervous system functions, the things that we were just talking about, sensory, motor, integrated functions. We can essentially call them the wiring of the body. And they're described as being extremely long living, living for over 100 years in cases when people do actually survive that long. They're amitotic, which means that they don't go through mitosis, which mitosis is cell division, where two cells come from one. So they don't split in half and two cells come from the original one, which explains why, why they need to live so long, because we have what we have and we don't make more, essentially. They have a high metabolic rate, which means that they use a lot of nutrients, oxygen, glucose especially. They also produce a lot of waste, and it explains why places like our brain need continuous blood supply. If they don't, we don't survive very long without that blood supply. Some neurons are tiny and propagate impulses over a short distance, strictly within the central nervous system. Others can be the longest cells in the body, traveling from like places in our spinal cord all the way out to the tips of our toes. In order for signals to travel, they need to do so at really rapid speeds, and that can range as slow as a half to 130 meters per second, which you may know, but I definitely think you would know what 1 to 290 miles per hour will be. So for reference, that's incredibly fast. Okay, as far as neurons go, there's kind of two main parts to it. What we describe as the kind of control center of a neuron is its cell body, which is typically where you find, I shouldn't say typically, it's where you find the nucleus. You find the same organelles that we've seen in other cells, mitochondria and the rough endoplasmic reticulum and smooth endoplasmic reticulum. You find lysosomes, all those wonderful things that we've learned about previously. You can see some of those are labeled here. These things called mesobodies are just a different way to describe the RER of the cell body. Usually the cell bodies of neurons are located strictly within the central nervous system. If we have a group of neurons, the cell bodies typically cluster up together. A singular one of those would be called a nucleus. Multiple of those would be called nuclei, because that's how you pluralize nucleus. And we've already seen the term ganglia here today. Ganglia are clusters of cell bodies of neurons in the peripheral nervous system. All right, extending off of a neuron are what we call nerve fibers, and there's typically two types. A whole bunch of these things that look like bare tree branches, which are called dendrites, and then a long, relatively unwinding axon, which is like the wire of the cell. And dendrites are the primary site for receiving signals from other neurons. Once again, they receive bare branches of trees extending from the cell body. Plasma membrane of dendrites and cell bodies too contain numerous receptor sites for binding chemical messengers that are released from other cells. So when a signal comes into a neuron, typically it's going to activate and it's going to involve these dendrites. Okay? An axon, as I said, is relatively unbranched, and it conducts nerve impulses to points far away. In cases of certain situations, it can run up all the way from the tips of your toes to your legs and into your spinal cord. So talking super, super long. An axon often joins the cell body at a kind of cone-shaped elevation called an axon hillock. Located in there is what we call its initial segment, which is the part closest to the axon hillock. Not shown here would also be what's called a trigger zone. The junction of the axon hillock and the initial segment where nerve impulses arise. So it's exactly what it sounds like, where an action potential is triggered, and then from there it's just going to spread all the way down the axon to the very end. In some cases, axons do have kind of right angles branching off. We'll call these axon collaterals. We show one here. What's not illustrated in this illustration would be the fact that an axon is going to continue going off the same direction, just like it is here. So all this stuff, including these branches, would be going off in that direction as well. Talk about those branches here in a second. The axoplasm is a fancy name for the cytoplasm of an axon. Axolemma is a fancy name for the plasma membrane of an axon. You can see those things are labeled down here. Let's talk about those branches at the end. The very tip of the axon, it branches kind of like dendrites, except we call these axon terminals, also known as telodendria. And I'm kind of zooming in to show that at the very tip of each one of those, they kind of have a little fattened structure called a synaptic end bulb, also known as a synaptic knob, which is where we store and release what we call neurotransmitters. So the chemical messengers that each neuron will release in order to activate another neuron or activate a cell, like a muscle cell or a gland cell, that kind of stuff. Now this neuron happens to be myelinated, which means it's covered in what we call a myelin sheath, produced by cells called Schwann cells that we'll talk about. Not all myelin is formed by Schwann cells. We also have another type of cell that creates the myelin sheath around cells, around the axons. We'll talk about those here in just a minute, a couple slides from now. One key thing I want to point out before I move on from this picture is that this kind of illustration is a little bit misleading because it kind of looks like this whole segment right here of the axon is not covered with myelin. They've just kind of removed it so you can see the axon a little bit better. But there are segments where there is no myelin covering an axon, which those are called nodes of Ranvier. And they enable a action potential to skip from node to node like a rock skipping on water, which allows for very rapid nerve signaling. And that's why those signals can travel at speeds like 290 miles per hour. So, pretty amazing. Over on the right, this is a picture of, you can definitely see this, the cell body of the dendrites. There's the axon of looks like a motor neuron, and it looks like they're zoomed in 400 times what the naked eye could see. So pretty amazing. A lot of similarities, and for good reason, to what we're showing over here on the left. Very, very important picture, I would say. Okay, what I want to do on this next page is talk about the point where one neuron ends and another begins, what we call a synapse. We can describe a synapse as the point of contact, and I would use the term contact with air quotes because it's not actual contact between two neurons. It is where two neurons communicate by way of chemical messengers known as neurotransmitters released from the synaptic end bulb of one neuron and then travel across, and this illustration doesn't show it, but the dendrites would be where those neurotransmitters are going to go on the second neuron. The first neuron is always called a presynaptic neuron. The second neuron is always called a postsynaptic neuron, and that's because pre means before and post means after. Now, what's key here to understand is that we don't always just have neurons communicating with other neurons. It can also be effector cells. So maybe instead of a neuron here being a postsynaptic neuron, it would be a postsynaptic muscle cell causing the muscle to contract, which means that the presynaptic neuron would release neurotransmitters that would then bind to muscle cells instead of neurons. Okay. Now, let's talk about the synapse anatomy down here at the bottom a little bit more. Looking at this illustration, which is from your textbook, we show one singular synaptic end bulb, and it's not labeled, but if you wanted to, you could just kind of draw a bracket over here off to the right and label this as the synaptic end bulb or knob. Once again, is a tiny bulge at the end of the terminal branch of a presynaptic neuron's axon, and it's embedded with voltage-gated calcium channels, what these little purple things are. When action potential arises at each of those knobs, calcium rushes in and triggers the movement of these little bubbles. Those bubbles are called synaptic vesicles. And the whole structure is a vesicle. And inside, we see these little red dots. You can see some more of those, which are the neurotransmitters, those chemicals that are released from what we call the presynaptic membrane. There's the presynaptic membrane all the way across, okay, into what's called the synaptic cleft, which is this space right here between what we call the presynaptic membrane and this, which is the postsynaptic membrane. Okay. The illustration does its best. It's not the most perfect illustration ever. This is not an actual point of contact. As I said, I would use air quotes to describe contact because they're not actually touching. It's almost right on top of one another without actually touching. And I always kind of think of little kids or siblings when one's holding their finger to the other person's face close as they can without touching, and they say, "I'm not touching you, I'm not touching you." It's kind of what it is. They're so close, yet they're not actually touching. So the space between them would be the synaptic cleft. This would be the presynaptic membrane, and this would be the postsynaptic membrane. And the postsynaptic membrane is typically going to be, once again, the dendrites of that postsynaptic neuron, because that's where you find a lot of those receptors for the neurotransmitters. All right, let's talk about the ways to classify neurons. There's two kind of major ways to classify neurons. We'll talk here first about the structural classification, which is based on their kind of, well, obviously structure. Diversity as far as neurons goes, pretty great for size and shape, especially when it comes to the way that the nerve fibers, those extensions, especially the dendrites, branch off of the cell body. The most common type of neuron in the body, and that's what was shown not only here but on that previous page, was what we call or is a multipolar neuron. Multipolar neurons are known as multipolar neurons because they've got multiple extensions, a whole bunch of dendrites, and at least, well, one axon. So it's at least 3, probably more like hundreds when you calculate all those dendrites up. Unipolar neurons, I'll talk about those here on the right. Uni means one, the cell body's off to the side. There's one extension connected to it, one extension that then forks to go to the dendrites on one side and the axon terminals on the other. Okay. These are the ones that are going to carry signals into our brain and spinal cord for, sensations of like touch, pressure, pain, temperature. So for what we call our general sensations. The most rare of our structural classification neurons are called bipolar neurons. And we all know bi means 2. There's one and a second extension with the cell body being located in the middle. These are very rare, as I mentioned, only found in specialized sensory organs like our eyes, our ears, and in our nose. So if you've ever heard of like rods and cones are this type of neuron. There's even actually neurons in our eyes called bipolar neurons that are also obviously bipolar in structure. So we don't call them rare as in some people have them and others don't. They're rare just because there's not a whole lot of them in our body compared to what we call the unipolar, and by far away the multipolar. All right, at the bottom here we show the functional classification neurons, which is based upon the direction in which they carry their nerve impulses relative to the central nervous system. First type I'll talk about are the sensory neurons, which both these two, both bipolar and unipolar, can be. More than likely, is going to be unipolar, which is what we're showing here, carrying signals into the central nervous system. And in this case, maybe that signal is carrying information for the sense of touch or pressure or itch or tickle, whatever. But that signal is going to come into the central nervous system. And we find these in what we call the sensory ganglia outside our central nervous system. I'll talk about interneurons next, which are located between the sensories and the motors. And that's why I like the term interneurons, because inter, as we know, means between. These guys are going to be strictly found inside the central nervous system, which is why they're the most numerous of our entire body. Of the neurons in our body, 99% of them are this type. Another way to describe these are association neurons, but as I've told you, I like the term interneurons a lot better. Now we can actually classify them as motor or sensory, which relates to kind of which direction they're sending the signals inside the central nervous system, like up the spinal cord or down the spinal cord. Notice their structural classification are the multipolar type, as are the final type, what we call the motor neurons, which have their cell bodies located in the central nervous system, and then the axon extends out through the peripheral nervous system and eventually out to the effectors, where they then can cause things like muscle contractions or secretion from glands. As far as they go, another term for those are efferent. Same thing we've learned earlier. Those terms kind of get used synonymously. Same thing for sensory or afferent. Okay, so that's the neurons of the body. Kind of switch gears here and now talk about supporting cells of the nervous system, what we call neuroglia, or glia for short. Non-excitable, which we learned the term excitable means that they can generate action potentials. These guys don't do that. Instead, they're supporting cells. They do make up a very large very large portion of our central nervous system, half the volume. And speaking of the central nervous system, there are 4 that are found in there, and that's what these 4 here are listed on this page. That means there are 2, since there's 6 total, that are strictly found in the peripheral nervous system. We'll talk about those on the next page, but first let's talk about the 4 that make up the central nervous system. First type I want to talk about are called astrocytes. These kind of purplish, bluish ones in our figure are supporting cells that wrap around blood capillaries in our brain and spinal cord and create what's called a blood-brain barrier, or BBB for short. It's exactly what it sounds like. A barrier to prevent certain things from leaving the blood and entering into our brain and spinal cord, to prevent things like toxins and drugs from interfering with those activities of our neurons that we've been talking about. Notice here we can see some that are just, wrapping right around this blood vessel right here. Now, do they allow nothing through? No, sure. They're going to allow certain things, nutrients and waste that need to leave the brain and spinal cord have to pass through the blood-brain barrier. So astrocytes help with picking up those nutrients and moving them along, either to the delicate cells to the brain or away from them. Some astrocytes actually do support neurons due to their considerable strength, help maintain the appropriate chemical environment for the generation of nerve impulses, and may also actually even help in learning and memory by influencing the formation of those synapses that we were just talking about. You haven't noticed yet, the picture actually at the bottom shows 2 types, protoplasmic and fibrous. The difference relates to where they're found. Protoplasmic, we'll talk what gray matter is here in just a second, whereas fibrous are found in what we call the white matter. Another difference is these are going to have short branching processes, whereas fibrous are going to have much longer unbranched processes. Microglia are these kind of tiny little green cells, which are more like white blood cells in that they are immune in function, responding to areas of infection, trauma, or even a stroke, where they function as phagocytes, which means that they kind of act like Pac-Man by performing phagocytosis, chewing things up, make cell debris, microbes, damage nervous tissue, and digesting them, which is pretty cool. Ependymal cells are these down here at the bottom, have a lot of similarities in shape and structure to epithelial cells that we learned about earlier in the semester. These happen to be kind of cube shaped. They can be a little bit columnar in shape. A lot of them actually have cilia or microvilli, which enables them not only to secrete, but also to circulate what we call cerebrospinal fluid. Cerebrospinal fluid is fluid that's found in cavities in our brain and spinal cord. This happens to be what we call a ventricle, which is a chamber. We have 4 of them in our brain where we find cerebrospinal fluid. We'll talk more about cerebrospinal fluid when we get into our next couple lectures. And the final type of cells have a fun name to say, oligodendrocytes. They kind of, to me, always look like an octopus. And they are going to extend around and form the myelin sheath that wraps around axons of our neurons. And so I've already kind of mentioned a little bit about myelin, but myelin is a multi-layered lipid and protein covering that insulates axons from one another and increases the speed of nerve impulse conduction. A key feature of oligodendrocytes is that a singular oligodendrocyte can form the myelin sheath around not only one, but multiple axons, which is different than what we'll see on this page with what we call Schwann cells. Schwann cells and satellite cells are strictly found in the peripheral nervous system. Schwann cells shown here at the top in our 3 images, forming the myelin sheath around axons in the peripheral nervous system. The myelin sheath is still the same, fatty insulation, helping to protect the nerve fiber, electrically insulate them from one another, and to increase the speed of nerve impulse transmission. Unlike oligodendrocytes, which can myelinate several axons. Each Schwann cell myelinates only a singular axon. So what that means is, and we can see that here in letter A, this kind of pink blob right here is a single Schwann cell. Inside of the cell is the myelin sheath. Back here would be another Schwann cell, and inside would be its myelin. And back here, a third Schwann cell would be creating the myelin around this portion of the axon. Okay. Notice we can still see the same features of Schwann cells what other cells have, which includes a nucleus, a cytoplasm, so on and so forth. What's cool about Schwann cells, they have a special name for their outer wrap. We call it neurolemma. A very important feature of the neurolemma is to allow for the regeneration of injured nerve fibers. And that's something unique to peripheral nervous system neurons that central nervous system neurons don't have. If you think about it, typically things like brain and spinal cord damage are permanent, which means, someone's paralyzed, they're not typically going to get that ability to walk again or whatever. If you have nerve damage in your arms or legs, not involving the spinal cord or brain, there's a good chance that can be repaired, and that has to do with the neurolemma that I've been mentioning. If I go back to this picture, they actually show the neurolemma and the myelin inside, the cytoplasm, and so on and so forth that we've been mentioning here. I mentioned earlier in that same figure about what we call nodes of Ranvier, which is that microscopic gap located between places where there is myelin surrounding the axon, which once again allows an impulse to skip like a rock on water. We call this saltatoric induction from uninsulated portions to the next. Now, Schwann cells can also be found associated with unmyelinated axons. Not all axons have a myelin sheath, and the Schwann cells that are involved with those just basically encircle those and kind of just separate them from one another. Can do as many as 20 or more unmyelinated axons. On the far right here, we're showing a group of satellite cells. Inside of this kind of purple cluster, you can see in yellow would be the neuron, its cell body specifically. And we can see the function of satellite cells is just to wrap around the cell bodies of neurons to help exchange materials between the cell bodies and the interstitial fluid. So not a super important thing, but as far as the image goes, but still important functionally, obviously. Down here at the bottom, we show on the left side how myelin is formed, which is kind of cool. These are pictures from a microscope, zoomed in tremendously 25,000 times to show the function of those Schwann cells and creating myelin surrounding, in this case, a couple different axons, and over here on the right, several unmyelinated axons. We don't see that thick kind of dark burgundy red around the axons over here as we did over here. Still, both of them are involving Schwann cells, as you can see. All right, and then to kind of wrap us up here, let's just talk about collections of nervous tissue and the components, some of which we've actually talked about. So reading through, components of nervous tissue are grouped together in a variety of ways. Neuron cell bodies are often grouped together in clusters. We've mentioned ganglion, ganglia, nuclei or nucleus. Axons are kind of the same way. They group together instead of in clusters as in bundles. Okay. So let's talk about ganglia and nuclei here. Ganglia, plural form of ganglion, is a cluster of neuronal cell bodies located in the peripheral nervous system, and as we've mentioned several times, closely associated with cranial and spinal nerves. Nuclei is the plural form of nucleus, cluster of neuronal cell bodies in the central nervous system. Talk about nuclei, especially when we talk about the brain. Nerves, like cranial and spinal nerves, are bundles of axons, specifically found in the PNS. Okay, and as I've talked about, cranial and spinal nerves associated with the brain and the spinal cord, respectively. A singular nerve typically is going to be made up of hundreds of thousands of nerve fibers. So it's not like you have one singular neuron per nerve. It's anything but that. And when we talk about those neurons, remember, a lot of times you'll find the dendrites and cell bodies of those neurons located back in the central nervous system, like we showed here, and then the axons extending out in the peripheral nervous system. Tracts, we'll talk a lot about tracts when we get to the spinal cord and brain, call these bundles of axons in the central nervous system, allowing for interconnection between places within the brain and spinal cord. Also, we have widespread regions of nervous tissue that are grouped together as either what we call gray matter or white matter, based on their appearance when dissected. And when we're talking about dissecting, we're talking about either the brain or the spinal cord. And they call them gray white matter because of what they actually look like. Here we can see brain, and this is a dissected section of the spinal cord. It's not as very vibrant here as it is over here, but the white matter literally looks white. The gray matter is a lot duller. Maybe it's not necessarily a nice gray color. It's more like a dark tan, maybe. But the reason that white matter and gray matter exists is because of myelination or lack thereof. Myelinated fibers within the brain and spinal cord are what elicit that white matter. In the spinal cord, it's on the outside, and in our brain, it's on the inside, the deeper portions of our brain. And that's the case for what we call cerebrum, and then the other big portion of our brain, what we call the cerebellum. On the outside of our brain and on the inside, kind of forming a capital H shape, is what we call the gray matter, which is because, or appears gray because of very little to no myelin. And instead of having myelinated axons, you've got the cell bodies of neurons, their dendrites, the axon terminals, or even just bundles of myelin axons and neuroglia. So, all right. Well, that is it. So I will call it there. Thank you guys. I'll see you later.